Hi, my name is Peter Flood. I'm a third-year PhD student at Florida International University, and thanks for clicking into my talk. Everglades National Park, where my research takes place, is no stranger to non-native and invasive fishes, as you can see from the stats listed on this slide. And the impacts of many of those invasions are still unknown as of yet, because there are many challenges, as we know, to understanding those impacts, and one of the major challenges can be the rarity of pre-invasion data. Luckily enough, I was able to inherit a pre-invasion data set of stomach contents and stable isotopes, which I'll be presenting data from today. My dissertation as a whole focuses on comparing the historic pre-jewelfish invasion, the bright red fish on the top right corner of the slide, food web, the pre-invasion food web, to the post-invasion food web in a contemporary study that we've conducted. And then we'll be comparing the metrics along this dividing line from top right to bottom left in the pre and post invasion settings. But today I wanted to take these data in a different direction. Specifically, I wanted to use these data to test both a hypothesis about invasions, the biotic resistance hypothesis, as well as using the niche breaths I have based on stomach content data from the pre-jewelfish invasion food web, and use those to test a broad hypothesis in ecology, such as the niche variation hypothesis. I was interested in testing both invasion and broad ecological hypotheses with the same data set. We'll start with the biotic resistance hypothesis, which states there's an inverse correlation between native diversity and invasion success, so a highly diverse ecosystem, such as the coral reef on the left, would be more difficult to invade, whereas a system such as the one pictured on the right may be easier to invade due to available niche space. So according to that hypothesis, the areas on this figure in bright red that are more diverse should be harder to invade, and the areas in the cooler blue colors should be easier to invade. Now, if we zoom in on Florida, and specifically South Florida, and this square approximates the Everglades National Park boundaries, and you can see that native diversity there is quite low, and the non-native diversity is relatively high. So we might expect that the biotic resistance hypothesis had a part to play in this. Next, I was interested in using my stomach content data to calculate the niche breaths and therein test the niche variation hypothesis that states that competition and niche breadth are inversely related for populations. As you can see in this figure on the right, in the presence of trout, the total population niche breadth of the other species of interest decreased. But I was interested in applying this to communities. For communities, we might expect that competition and total community niche breadth would be directly related. So I have a series of figures here on the slide to visualize that. As you move from lower competition to elevated competition, including increasing species richness, you start to see individual population niches will shrink, but the overall community niche may expand, and if it doesn't expand, we would at least expect to see increases in metrics of dispersion, such as mean distance to centroid for each population, from the population centroid to the community centroid, and metrics such as nearest neighbor distance, or the standard deviation of that. The Everglades is home to a variety of habitat types, and today I'll be talking about three. The pond, or alligator pond, that you can see on the left, the spike rush marsh, where I'm collecting a sample out there with another graduate student, and then sawgrass marshes that are off to the right. Each of these habitats has a distinct difference in species richness that changes across seasons between the wet and the dry season, and also the relative level of competition within the habitat that changes between seasons. My interest was in testing the biotic resistance hypothesis at a smaller interhabitat scale within the Everglades, specifically if seasonal changes in species richness led to seasonal changes in invasive richness, and if there was a correlation between native and invasive richness among habitats. To test the niche variation hypothesis, I was interested in asking if habitat season levels with higher competition have larger community niche areas, and if they show increased diet partitioning. So as I mentioned previously, this is a historic data set. The field sampling was exhaustive using all available methods, and it was stratified across the pond, spike rush marsh, and sawgrass marshes that I described earlier. And then in the lab, stomach contents were analyzed volumetrically and numerically. And then today, I'll be presenting the numeric data, and fishes were separated and categorized by species size class to account for ontogenetic changes. Originally, the stomach content data had over 300 prey categories that were qualitatively condensed down to 34 broader categories. I condensed these down further using an NMDS with k equals 2 to make them a two-dimensional data set. I then used the cyber package in R that was created to model niche areas, ellipse areas based on stable isotope data to model these two NMDS axes in place of isotopes to calculate probability distributions of those ellipse areas. And then I could calculate the probability that one ellipse was greater or less than another in size. And then I also calculated the Lehman metrics. And because of the probability distributions of the Lehman metrics as well, all of these were calculated using a Bayesian approach. I could make direct statistical comparisons among groups. This brings us to our first hypothesis about the biotic resistance hypothesis, and with that, I'll move into my results about the biotic resistance hypothesis and our first question there. In this blue box at the top, I have reported the wet season invasive species richness. There were 5 species found in the wet season relative to 26 native species, whereas in the dry season, there was a decrease in invasive species richness to 3, but also a decrease in the native species richness to 19. So it seems that although there was a decrease in native species richness from wet to dry season, that didn't result in an increase in invasive species richness from wet to dry season, as we might have predicted. Now with that, we'll move into the second part of the biotic resistance hypothesis for this talk. As you can see, what we found was a direct correlation between invasive and native richness. The invasive taxa are pictured on the slide, and the numbers represent the native richness in each of these habitat types. And you can see that those were directly correlated as opposed to negatively correlated as would have been predicted by the biotic resistance hypothesis. Next, we'll switch gears to the niche variation hypothesis, seeing if increased competition drove increased community niche areas. So those natural competition gradients within each habitat, moving from the wet season to the dry season, go in the direction listed here. In the sawgrass marsh, as the water levels drop, there's an increase in terrestrial prey that are available. And because this habitat is already more difficult to persist in and be successful in, fewer fish species are there to begin with, so this increase in prey availability decreases competition. In the spike rush marsh, there's a decrease in water level, there's a decrease in resources available, which means there's an increase in competition, there's fewer resources to go around. Whereas in the pond, the opposite story is true. In the dry season, there's an influx of these small fishes, which provides increased resources to the majority of the pond residents, decreasing competition. Now, the Lehman metrics, as I mentioned earlier, are metrics of dispersion fundamentally in space. So As I went over in the introduction, as competition increases, we would expect the community total area, the total niche area of the community, to increase if there are new resources being used that weren't previously because they're subpar or not as high quality, or it would stay the same if there are no new resources being consumed. Centroid distance, the mean distance from a population mean to the community mean, we would expect to increase. At nearest neighbor distance, we would also expect to increase as competition is driving a divergence of population niches within the community. And likewise, we would expect the standard deviation of that to increase as would be more specialization within a community as competition increases. On this slide, I have a box plot of the posterior distributions for total community niche area on the y-axis among habitat season levels, which are on the x-axis. The groupings, denoted by letters at the top of the graph, were created at a 5% threshold. For example, sawgrass during the wet season, the first distribution on this graph on the left-hand side, has greater than a 95% chance of being larger than the second distribution on this graph, sawgrass during the dry season. This result aligns with our prediction that the higher competition in the sawgrass during the wet season would result in a larger community niche breadth. We do not see this pattern in the other habitats at the 5% threshold. However, there is a change present in the expected direction for both habitats. Recall that in the spike rush marsh, competition increases in the dry season relative to the wet season. And here we see a marginal increase in community niche area for the dry season relative to the wet season. Similarly, in the pond, competition decreased in the wet season because of the influx of resources, those small fish that get concentrated there as the marsh dries. And we see here a decrease in total community niche area for the pond in the dry season relative to the pond in the wet season. For both the spike rush marsh and the pond, the seasonal differences occur at a 25% threshold. That is to say, there's a 75% chance the community niche is larger for the dry season pond community relative to the wet season pond community. Finally, for this figure and figures to follow that are very similar, while there are some statistical interhabitat differences or interhabitat season level differences at a 5% threshold or another threshold that I've denoted, I'll refrain from addressing those here because it's difficult to predict the relative amounts of competition among habitats because of other factors that could contribute to niche area among habitats in addition to competition. And now we'll move into the second part of the niche variation hypothesis in this talk, asking how competition impacts diet partitioning or those other metrics of dispersion in niche space. Now on this slide is another graph very similar to what I presented for total niche area. However, this is centroid distance, which as I described earlier is the average Euclidean distance of each species size class to the centroid of that community habitat season level. So if you recall, we predicted that this metric would directly correlate with competition. So higher competition, higher degree of trophic diversity because the competition is driving niche divergence, which leads to the community having an overall higher degree of trophic diversity. So as you recall, this first set of habitats here are the sawgrass. We predicted lower competition in the dry season, which should lead to a lower degree of trophic diversity in the dry season in sawgrass, which is what we see here. And again, as before, these thresholds are at a 5% level. Where before we only saw a difference at a 5% threshold in sawgrass, for centroid distance, we also see a difference at this threshold in the spike rush marshes. You can see between spike rush in the wet season and the spike rush in the dry season, there was again a difference at a 5% threshold. There was an increase in trophic diversity and centroid distance from the wet season to the dry season, which corresponds with our predictions from the hypothesis because of the increased competition in the spike rush in the dry season. Now in the pond, similarly to the total niche area, we do not see a change at a 5% threshold between wet and dry season. And this time we actually see the trend is in the opposite direction of what we would have predicted. Now, there being more changes at a higher threshold for the centroid distance is not surprising. As I stated earlier in the introduction, the total niche area for a community may not change because for that to change, new resources would have to be exploited. So in instances when that doesn't change, it could be because new resources are not being exploited, but the degree of trophic diversity is increasing because the relative proportions of resources being used by a species are changing and they are diverging from one another, driving an increase in trophic diversity. Here's another set of box plots, this time of nearest neighbor distance. Again, this is the mean Euclidean distance to each species size class's nearest neighbor in NMDS biplot space. This is a measure of trophic redundancy, but it can get a little confusing because nearest neighbor distance is inversely related to trophic redundancy. So higher nearest neighbor distances mean lower trophic redundancy and vice versa. Now we predicted that competition would increase nearest neighbor distance, thus decreasing trophic redundancy. Now for this figure, and the figure on the following slide, the groups are denoted at a 20% threshold because none of the changes occurred at or below a 5% threshold. However, these changes are likely still ecologically important. Now for the sawgrass, we predicted that lower competition during the dry season would decrease nearest neighbor distance, therein increasing trophic redundancy, and that is exactly what we see here in this figure. For the spikerush marsh, where we predicted the increased competition in the dry season would increase nearest neighbor distance, we did not see a notable change between seasons for the spikerush marsh. However, in the pond, we saw an increase in nearest neighbor distance in the dry season. This is the opposite of what we expected since competition is lower in the dry season than the wet season for the pond. This may be driven by the fact that while there's an influx of resources that lowers competition, those resources are themselves not very diverse, leading to an increase in trophic redundancy. Okay, so I promise this is the last set of box plots you'll have to look at during this talk. This one is of the standard deviation of the nearest neighbor distance. This metric is a measure of the evenness of trophic niches. Lower values suggest a more even distribution of niches, while higher values suggest increased specialization or increased diet selectivity among community members. We predicted that increases in competition would drive niche divergence, increasing diet selectivity. And again, these groups were made with a 20% threshold like in the previous slide. For the sawgrass marsh, diet selectivity decreased in the dry season under low competition as predicted. However, in the spikerush marsh, we saw a trend in the opposite direction of what was predicted. During the dry season, when competition in the spikerush is higher, diet selectivity decreased. This may be the result of diminished resources. There's a documented shift to a browner, more heterotrophic food web in the dry season in spikerush marshes. This increased importance of detritus may be driving a decrease in diet selectivity. There are simply fewer options on the menu to choose from. In the pond, we saw no change in diet selectivity between seasons. Now, another explanation for seeing trends that we did not expect, or trends that are the opposite direction, or no trend at all. This could be because we're only measuring a single dimension of the animal's niche, the trophic niche. Changes in competition across this natural gradient of habitat season levels could be causing changes in the animal's total niche, even in the predicted directions, but we wouldn't be seeing that because we've only quantified the animal's trophic niche, not the animal's total niche. In conclusion, for the biotic resistance hypothesis, we didn't find what we expected, and this is likely because different habitats across the marsh have different resource availabilities. There's some evidence that the pond, for example, is nutrient enriched compared to the spikerush and the sawgrass marshes, and this could be driving increased available niche space in the pond relative to those other communities. So while the pond has increased native species richness, it likely also has an increased available niche space because of those elevated nutrient levels. When we looked at Total area, we saw that the trophic niche area increased across a competition gradient in the predicted direction. Although this didn't always happen at a 5% threshold like it did in the sawgrass, it did happen at or below a 25% threshold in both the spike rush and the pond habitats, which is likely Finally, when we looked at the other layman metrics, for looking at diet partitioning among habitat season levels and across that natural competition gradient, we had mixed results. For some habitats, for some metrics, we had the predicted changes, in others we had no change, and in yet others we had the completely opposite trend as what we predicted. And this could be for a number of reasons, such as resource availability and other variables that we didn't consider. Going forward, it'll be important to consider additional variables concerning this work and how they are impacting both competition and relative niche area for populations and communities. Resource availability, such as elevated phosphorus levels, nutrient levels of one habitat relative to another, or predation, which has been shown to increase diet selectivity, could be influencing the results of this work. Going forward, I'd like to regroup these communities and populations by trophic yields to see if there are emergent patterns there. I'm interested in how stomach content niche breaths correlate with stable isotope niche breaths or fatty acid niche breaths. And then, of course, there's the current post-invasion jewelfish study, which we're working on right now and processing samples for. And then all of these trends that I presented in the historic community today will be able to compare to the post-invasion study among other response variables that we have. And with that, I would like to acknowledge my advisor, Joel Trexler, and the rest of my committee members for their feedback on my work throughout my dissertation. Bill Loftus, who collected these before data and did a lot of that heavy lifting on the original round of analyses. Bradley Strickland, who's another graduate student who I've collaborated with on heavily. Sarah Cisco, Maria Sabando, and Venus Garcia, who have done amazing work processing these laboratory samples. And then, of course, I would like to acknowledge all of our funding sources. Finally, I'm happy to take any questions, including those from right field.